Well, I'd like to thank the organizers for the invitation to participate in this session, and also the previous two speakers who gave an outstanding talks and introduction to the concepts of HIV clinical and virologic reservoirs, and so I will spend very, very little time on background slides. So probably my only background slide, why do current drugs fail to eradicate HIV? I think it's quite obvious, particularly to the people in this room. HIV establishes a latent reservoir in long-lived CD4 T cells during acute infection, and the viral reservoir is not eliminated by antiretroviral drugs that target replicating virus. Early initiation of ART and latency reactivation alone will likely not eliminate the viral reservoir, from evidence from a large number of groups, including the two previous speakers in this session. So there are multiple strategies for HIV eradication, and I won't go through these all, but I will concentrate today on some of our recent thoughts regarding the last two, which are immune-based interventions. I'll show you some very preliminary data on therapeutic vaccines, as well as more data on broadly neutralizing antibodies. So we showed in a paper about 2 years ago that if we use the monkey SIV model, then very early initiation of ART following SIV infection, even when we start as early as 3 days into infection, this is prior to viremia, so theoretically at an earlier point in time than any clinical trial could be done, and we suppress virus completely for a period of 6 months and we stop therapy, then we see 100% viral rebound. So basically this suggested that at least the initial seeding of the viral reservoir occurs very, very early within the first few days of infection and is refractory to even a prolonged period of ART. And the data on the viral rebounds that Brigitte Autran presented earlier in this session, even when humans are treated for 5 or more years, is certainly consistent with these data. So these data beg the question that other interventions are needed. So the first question that I will try to address today is, can therapeutic vaccines augment host cellular immune breadth and lead to immunologic control of the HIV viral reservoir? I'll show you some data that they can indeed augment cellular immune breadth in the non-human primate model, and we don't yet have data on whether this has an impact on the reservoir or viral rebound. So the vaccine that I'd like to introduce you to is an ad vector prime followed by an MVA boost vaccine that we shows at least a partial degree of efficacy as a prophylactic vaccine in Rhesus monkeys. In a more recent study, we explored whether the same vaccine might have therapeutic potential in acutely ART-suppressed SIV-infected Rhesus monkeys. So in other words, in an animal model where we can control the virus sequence at input, the timing of antiretroviral initiation, and the relative sequences that are used. So this study used 36 Rhesus monkeys that were infected intrarectally with SIV-MAC251. ART was initiated on day 7 of infection, which is during the upstroke of exponential viral replication and prior to peak viremia, using ART that we and others have proven to be fully suppressive in this model that involves a pre-formulated combination of tenofovir, emtricitabine, and dolutegravir. 18 animals received the vaccine, and 18 animals were sham controls. And the vaccine was given the Ad26 prime, expressing the inserts on Vgag and Paul, were given at weeks 24 and 36, so essentially 6 months of ART suppression prior to vaccination. And then the animals were boosted with MVA vectors expressing the same inserts at weeks 48 and 60, and at week 72, we have just discontinued ART. So the viral suppression is shown here with ART. You can see that on day 7 of infection, essentially during the upstroke of viremia, then virologic suppression occurs. Yeah, I can have my cursor. So virologic suppression occurs very quickly and effectively with the initiation of ART to undetectable levels in all animals. In both the sham and the vaccine groups, there's 2 or 3 animals that seem to take a little bit longer to control, but the vast majority of animals are controlled within a few weeks, and all the animals are controlled by 200 days. So you can see here the intervals of when we gave the Ad26 priming and the MVA boosting inoculations. So here are the T-cell responses in these animals, and we saw a dramatic immunogenicity of this vaccine, much higher immunogenicity than we see in a large number of studies we use these same vaccines in naive animals. So against either the vaccine-induced peptides, or the vaccine-encoded antigens, E543, or the current viral infection of the animals, SIV-MAC239, then we see a dramatic increase in the T-cell responses to ONVGAG and POL, as shown here essentially in groups 1 and 2, which were given the vaccine. So after the Ad26 vaccination, the prime, we saw ELISpot responses close to 2,000, and after the NVA boost, we saw responses in the 6,000 plus range, which are really very, very high. These residual responses actually are real responses and are those triggered by the infection alone. So there was essentially over a 100-fold increase in the antigen-specific T-cell responses. Perhaps more importantly, we also saw that this vaccine led to a dramatic increase in cellular immune breadth in these animals. These are individual animals prior to vaccination, infected animals on ART, showing an average of about 1 epitope in GAG. After the Ad26 priming, then it goes up to an average of about 4 epitopes in GAG, and after the MVA boosting, then it's about 5.5 epitopes in GAG. Similarly, if we look at on-subpool responses, then we go from an average of approximately 1 to approximately 6 epitopes, and for POL, we see a similar increase of about 1 to about 6 epitopes. So we see overall about a 6-fold increase in the cellular immune breadth compared to that generated by natural infection that's early suppressed if we administer this vaccine. And I should also mention, with this dramatic expansion of cellular immune breadth, then also the vast majority of epitopes induced by the vaccine actually were not primed by the virus itself, but are really new responses that are vaccine-induced. So, to briefly summarize this part of my talk, therapeutic Ad26 MVA vaccines are highly immunogenic in acutely ART-suppressed SIV-infected rhesus monkeys. The vaccine is substantially more immunogenic in the infected animals compared with our prior experience in naive animals, likely representing preserved T-cell help with early ART. We don't believe that it's expanding previously primed responses because these responses really were not primed by the virus. They're really primed de novo by the vaccine itself. There's about a 6-fold expansion of cellular immune breadth and over a 100-fold expansion of the magnitude of T-cell responses by vaccination. And we're just now assessing the impact of the vaccine-expanded cellular immune responses on potential viral rebound following ART discontinuation. But we do think that the ultimate potential of therapeutic vaccines might be enhanced when they might be co-administered with latency reactivation agents for a so-called shock and kill approach. So I'll turn now for the remainder of my time to a second immune-based intervention, which is broadly neutralizing antibodies. And can broadly neutralizing antibodies contribute to HIV eradication efforts? Several years ago, our group and others proposed that these should be explored more seriously. Potential clinical applications of broadly neutralizing antibodies for HIV could actually fall into more than just the cure arena. They can be used for primary prevention and pre-exposure prophylaxis, and there's actually one efficacy trial ongoing now with the VRC01 antibody to test that question. They could also potentially be used immediately following HIV exposure in humans as part of improved post-exposure prophylaxis regimens. They could be used for treatment strategies and potentially explored for cure or eradication strategies. The development of broadly neutralizing antibodies for clinical application really became possible over the last 5 years because of the generation of a very large number of impressive new monoclonal antibodies against HIV envelope. This is a slide that Dennis Burton developed showing the generation of antibodies from multiple different groups that are here in gray are the traditional broadly neutralizing antibodies for HIV that we had about 5 years ago. And over the last 5 to 7 years, now we have monoclonal antibodies that are 1, 2, and even 3 logs more potent while also maintaining substantial breadth compared with previous antibodies. So these now have the potency that they might be able to be viewed as pharmaceutical agents. So there's antibodies against multiple different targets on the HIV envelope surface. Imagine that this is the virus membrane. Here in light blue is an envelope protein, and these are the positions of different monoclonal antibodies for HIV. These actually reflect actual crystal structures, so this is actual real. The antibodies that are furthest along in clinical testing bind here to the CD4 binding site, as exemplified by VRC01 or similar 3BNC117 antibody. Antibodies that are either just started or soon to start clinical trials are directed against this V3 glycan loop on sort of the top corner of the molecule, as evidenced by PGT121 or 101074, and antibodies that will soon be in clinical trials potentially next year are antibodies against the very top of the envelope spike, against the V2 glycan region, as evidenced by PGDM 1400 or CAP256. We started to get interested in particular in PGD121, which is a potent and broad V3 glycan-dependent antibody, because there have been at least 4 or 5 different labs that have shown that it works very, very well for prophylaxis at very low concentrations. So it's an exceptionally potent antibody. And then about 3 years ago, we showed that this antibody also has substantial therapeutic effects in SIV-infected, untreated, viremic, chronically infected viremic animals, as shown by this summary figure. And in a group of 18 animals, there was a very classic response. A few animals that had very low-level response had immediate suppression and actually didn't rebound. But most animals that had starting viral loads between 3.5 and 5.5 logs had a suppression of virus to up to 3 logs within about a week, and then maintained undetectable virus for a period of time until antibody titers drifted to below about 1 microgram per ml, and then had a very classic viral rebound pattern. And a recent study that Michelle Nussenzweig's team published last year with a CD4 binding site antibody 3BNC117 actually recapitulates these monkey data and showed that a single administration of 3BNC117 led to about a 1.5 log drop of virus for a brief period of time. And this is about similar to what we see with this antibody in monkeys too. It's not nearly as potent as the PGD121 antibody. So it looks like the early data generated with monoclonal antibody therapeutics in humans, in HIV-infected humans, is very concordant with the data generated in the monkey model. But our published studies and those of others have shown that PGD121 administration can reduce circulating virus as well as proviral DNA in viremic SIV-infected animals. However, these studies did not directly address whether the antibody can actually target viral reservoirs, which can only be done in the setting of ART-suppressed animals. So we're currently modeling the efficacy of PGD121 plus ART, both in the setting of chronic SIV infection in animals as well as acute SIV infection in animals. So in a pilot study in chronic SIV infection, 9 monkeys were infected with SIV SF162P3 7 months prior to infusion, had baseline viral loads typical of that of previous studies, about 3.5 to 5 logs RNA copies per ml. ART was administered for a period of 20 weeks. And then group 1 received ART plus 4 infusions of PGD121, and group 2 received ART alone, and then we stopped therapy. So here is the schema of the experiment. You can see that both of these regimens, of course, not surprisingly, suppressed plasma viral loads to undetectable levels, and then we stopped therapy. And so what happened? So in the first few weeks, we actually were very excited because the ART alone animals, all 5 of these animals rebounded, whereas in these animals, two animals had a classic rebound pattern, and the other 3 animals appear to at least be early controllers. But with more time, then all of these animals in the PGD121 group also rebounded. But there were several animals, as shown here, that appear to have delayed rebounds. So it's certainly a suggestive, although perhaps not a definitive result because of the small animals numbers in this study. But we did notice that the time to viral rebound correlated with a baseline pre-ART plasma viral loads. And so the baseline viral loads prior to treatment appears to be a factor that determines how these animals respond to the subsequent immune-based interventions. So in this study, ART plus PGD121 delayed but did not prevent viral rebound. The delays correlated with reduced proviral DNA in PBMCs and tissues. I didn't have time to show you this. The time to rebound also correlated with baseline pre-ART viral loads. And these data at least lead to a suggestion that antibodies may have utility, but alone, at least in chronic infection, appear to be insufficient for HIV eradication, which is not a surprise. So to try to further show the potential of these antibodies, we did a study in acutely SIV-infected animals. Again, because we can time the interventions in a way that can't really be done in clinical trials. So we decided to introduce interventions on day 7 of infection, as well as an extreme case on day 3 of infection. We figured that on day 3 of infection, we know that the viral reservoir is largely seeded already, so we should get rebound in the most controls following ART discontinuation. But also, it's sort of at the brink, because if we start therapy on day 1 or 2, then some animals actually don't rebound as post-exposure prophylaxis, again, similar to humans. So we asked the question, if we intervene very early on, not just with ART, but also with an extraordinarily potent broadly neutralizing antibodies, then could we actually prevent or further delay viral rebound? So in this experiment, we infected 64 Rhesus monkeys with SIV SF162P3 on day 0, and then we initiated either ART or a single infusion of the antibody PGT121 either on day 7 or day 3. And these were essentially 2 parallel studies, each one 32 animals with 8 animals per group, in which we looked at no intervention, ART alone, PGT121 alone, or both combined. So to some extent, this is more of a post-exposure prophylaxis type study, but with the intention to see whether we could have any evidence for potential cures. So this is the pharmacokinetics of the antibody. The kinetics is actually relatively, the pharmacokinetics is relatively short in the context of acute infection, largely because we think that the huge amount of replicating virus is a sink and really absorbs a lot of the antibody. So the half-life is actually relatively short, and by 28 days, then the antibody is gone. So here are the results from the day 7 intervention. Here on the upper left, in the no intervention group, then this is essentially the natural history of SIV infection in monkeys. We reach a viral peak at about 10 to the 7th copies per ml on day 14, and then we go to a viral set point that's about 10 to the 5 that essentially goes on perpetually. If we look at the ART alone, then we see that between day 7 when we started and day 10, viral loads actually go up for the first 3 days in 100% of animals. Then, very abruptly, virus is controlled to undetectable levels. However, this is brief, and essentially within 2 weeks of stopping ART, 100% of the animals rebound. If we give a single infusion of the antibody on day 7, then we essentially blunt the further increase of viral loads. However, this alone, because of the vast amount of virus replication, presumably is insufficient to suppress virus. But nevertheless, on day 14, then we have a 3-log reduction of peak viral loads, which is about 10 to the 7th here, down to about 10 to the 4th here. So this degree of antiviral efficacy of about a 3-log effect is actually very consistent with our previous observations. But by day 28, then there's really no evidence of difference in viral loads between the antibody group and the no intervention group. If we look at the combination of these, then we do see evidence of a more rapid virologic control. As evidenced by day 7 to day 10, then the virus is already going down, and we also see some evidence of a delay of viral rebound. So viral rebound doesn't occur until a couple weeks later than in the ART alone group. But the important thing is that all animals still rebound. So this is just a summary of that data. This is the group medians showing the courses of the no intervention group shown in black, the antibody alone group in green, and the suppression and rebound seen with ART alone in blue, and the faster suppression in the combination group shown in red and quantified here, and the delayed rebound shown here in red compared with blue, and the delay was about 2-fold from about 10 days to about 24 days, so about 2.5-fold the rebound time. So clinically probably not significant, but this was a statistically significant result, perhaps one that can be built on. Here are the results when we intervene extraordinarily early on day 3. Essentially, the vast majority of these animals actually were aviremic at the time of initiation of therapy. So this is, again, the no intervention group. In the ART alone group, then we start ART, and 2 animals actually blip up from day 7 to day 10, perhaps reflecting a large amount of virus already replicating in the gut. And after ART is stopped, then all animals except possibly for 1 rebound. If we administer on day 3 a single infusion of the broadly neutralizing antibody PGT121, then we actually can ablate 7 logs of replicating virus during acute infection with 2 small blips here. And then by day 60, then the vast majority of animals rebound. although seemingly at a fairly large difference in time frames. And if we give both ART plus PGT121 on day 3, then there's no evidence of any virus during acute infection. And then when we stop the ART, then we still get rebound in the majority of animals. A couple animals we're still following. This is an actively ongoing study. However, the rebounds do occur later than in the ART alone group. So we were not able to cure any animals, or at least we don't think so, certainly not more than a handful. So overall, the study was not successful in eradicating the virus, but it did achieve an antiviral effect on the viral rebound. And this is, again, summary data showing the group medians, the untreated group in black, and then ART alone in blue, antibody alone in green, and combination of both in red, showing that there was a delay in viral rebound. Now, the rebound in the day 3 intervention was actually longer than in the day 7 intervention across the board, and we also saw about a 2- or 3-fold increase in the time to viral rebound comparing ART alone with ART plus PGT121. Also, I think as mentioned by the previous speakers, our virologic assays to measure these effects are insufficient. So we were actually unable to measure any level of viral DNA in PBMCs prior to viral rebound in all of these interventional groups on day 35. Then these are virtually all negative with a sensitivity of 3 copies per million cells. This was also true if we look in lymph nodes, then these are negative prior to rebound on day 35. And it's also true in the gut, negative in all 3 groups on day 35. And we know almost all these animals, if not all of them, eventually rebound. So as of this moment, with a DNA sensitivity of 3 copies per million cells, looking in gut, lymph nodes, and blood, we're unable to have a marker that predicts viral rebound. So in this study, compared with ART alone, ART plus PGT121 led to more rapid viral suppression and a statistically significant 2- or 3-fold delay in viral rebound following ART discontinuation. So these data, although this time frame of rebound is probably not clinically significant, it does suggest that the proof of concept that broadly neutralizing antibodies might be able to enhance virologic control that is achieved with ART and might be able to target at least the cells that are the source of viral rebound. I hesitate to actually call them reservoir cells because we haven't directly shown that, but we have impacted the presumptive impact of the reservoir, which is viral rebound following ART discontinuation. So PGT121 alone was comparably effective to ART alone when administered on day 3. I think that's pretty impressive. A single infusion of an antibody can achieve as much as 4 weeks of continuous antiretroviral drugs, but less effective when administered on day 7. All regimens initiated on day 3, even when it's prior to viremia, reduce viral DNA to undetectable levels in blood and tissues, but still fail to prevent reservoir seeding and viral rebound. And this confirms the earlier data we had showing early reservoir seeding in the SIV model. Now we showed in the SIV model as well, and suggests the refractory nature of the acutely seeded reservoir cells. So we do intend to move this into clinical trials, and we hope that our first-in-human PGT121 study in humans will start later on this year. And I'll conclude with just a few comments about antibody combinations. It's likely that a single antibody will not be sufficient for either prophylactic or therapeutic purposes, and so we're very interested in combining PGT121, which is a V3 antibody, with either CD4 binding site antibodies or V2 antibodies. I won't go through all the specifics, but here are the different locations again. We're particularly interested in combining PGT121 here with one of these antibodies in the V2 region for the simple reason that if you look at all the viruses, then there is a very good complementarity in terms of coverage of the V2 antibodies and the V3 antibodies. So you can see that most of the viruses that are missed by PGT121 shown here are covered very well by the V2 antibodies, PGDM 1400 and Cap256. And similarly, most of the viruses that are missed by the V2 antibodies are covered very well by PGT121. So, in fact, the combination of these 2 antibodies can achieve 98 to 99% coverage of global viruses. So, to summarize the antibody component of my talk, broadly neutralizing antibodies may have a role in HIV eradication strategies, likely as part of combination regimens. Again, we don't think that antibodies alone will be able to do the trick. PGT121 plus ART delays but does not prevent viral rebound as compared with ART alone in SIV-infected rhesus monkeys. The viral reservoir is seeded very early in both SIV and SIV infection and appears already refractory to elimination by day 3. The viral DNA assays, as I discussed, appear to be insufficient to predict viral rebound, and combinations of as few as 2 antibodies might be able to achieve robust neutralization coverage of global viruses. So just a few thoughts to conclude. We would propose, at least, that therapeutic vaccines and broadly neutralizing antibodies should be explored further as components of HIV cure strategies. These immune-based interventions will likely not be sufficient in their own right, that's my opinion, and will likely be most effective when used as components of combination interventions together with either immunomodulators or potentially latency reactivation agents. We would also propose that ART-suppressed SIV-infected rhesus monkeys represent a potentially useful animal model for testing and maybe comparing novel cure interventions or concept testing that might be able to facilitate and galvanize and accelerate clinical trial efforts. And we hope that clinical trials with our Ad26 MVA therapeutic vaccine, as well as the PGT121 antibody, will start before the end of this calendar year. Stop there and acknowledge all the people who are involved in this program. Dennis Burton at Scripps, or my lab at Beth Deaconess Medical Center, Dennis Burton at Scripps for the PGT121 antibody, our collaboration for vaccines with MHRP, as well as the Krusell-Janssen group, and most of the therapeutic vaccine work has been funded by the Gates Foundation. I'll stop there, and if there is time, take any questions.
Take one quick questions. Anyone? I have one question for you. So is there any way to measure the breadth and potency of cell-based immune therapeutic vaccines? For example, how do you measure the breadth coverage of CD8s?
So the crude way is essentially just mapping the epitope repertoires. A more sophisticated way is then to map those responses against the sequence of the autologous virus. For NHP studies, that's certainly possible because we know what the infecting virus is, although in chronic infection, of course, it does diversify. So I would think that to answer your question, then we would probably want to sequence autologous virus in each animal, and then on an animal-by-animal basis, then show not only reactivity against consensus peptide pools, but also the degree to which the actual virus is covered by those responses, and preferably the viral sequences in the reservoir cells and not just circulating virus.